Hi everyone, and welcome back to this NEET sessions, NEET biology practice sessions. Now today we are moving ahead with the fifth practice session, and we'll take another set of questions from the NEET paper of 2017. Okay, so let us move ahead and take a look at the papers. Now one thing that you need to focus, or one thing that you need to bear in mind, that a lot of questions have been repeated over the period of years that we have seen the question papers. Okay, so let us move ahead and take the questions one by one. So the first question is: the decrease in blood pressure or volume will not cause the release of what? Okay, so atrial natriuretic factor, aldosterone, ADH, and renin. Now, to understand this, what we need to understand is the renin-angiotensin mechanism, isn't it? Now, renin-angiotensin mechanism is responsible for what? Renin-angiotensin mechanism is responsible for increasing the volume of the glomerular filtrate, or increasing the urine formation, to increase the GFR, isn't it? That is what is renin-angiotensin mechanism. Now, renin-angiotensin mechanism works on the principle of elevating the blood pressure, right? So renin is secreted by what? Renin is secreted by the juxtaglomerular apparatus, isn't it? The cells of the JGA secrete renin, and renin, what does it do? It converts angiotensinogen to angiotensin 1, right? This angiotensin 1 is further converted to angiotensin 2. Okay, now angiotensin 2, angio, blood vessels, tensin, compression or tension. So what do you think angiotensin is going to do? It is going to increase the pressure on the blood vessels. So if the pressure is increased on the blood vessels, it causes constriction basically. So it causes vasoconstriction. So it's a powerful vasoconstrictor. So since it causes vasoconstriction, the blood pressure increases. And as the blood pressure increases, the GFR increases. Now, apart from this, we also have aldosterone mechanism, okay, which is responsible for reabsorption of sodium, okay, into the blood. So again the blood pressure increases. Yep, ADH is antidiuretic hormone, but its other name is vasopressin. Excuse me. So ADH or vasopressin is another vasoconstrictor. So again the blood vessel constriction is taking place, and ultimately what's increasing is the blood pressure. Now, because of this increase in the blood pressure, what is going to happen? The glomerular filtrate is going to be in more amounts, but this increased blood pressure is going to increase the pressure on the heart. And to now decrease this increased pressure, the heart secretes atrial natriuretic factor. So atrial natriuretic factor's role is what? Its role is to reduce the increased blood pressure, right? So if the blood pressure is already low, if there is already a decrease in blood pressure, will the atrial natriuretic factor be released? No. So a decrease in blood pressure or volume will not cause the release of what? Atrial natriuretic factor, right? Yep. So this is basically how the process works. This is how the component, or the atrial natriuretic factor, is going to work, right? Next, which of the following are not polymeric compounds? So let's understand this. Which of the following are not polymeric compounds? So let's go back to biomolecules. Yeah. Now, when we talk about biomolecules, recall that in biomolecules we have carbohydrates, yep, then we have proteins, we studied nucleic acids, and we've studied lipids, correct? Now in carbohydrates, what do we have? In carbohydrates, we have monosaccharides, which are simple sugars, disaccharides, which are formed by the combination of these simple sugars, right? And then we have polysaccharides, which are formed by repeating units of the simple sugars, like for example, starch, glycogen, cellulose, inulin, all of these, right? So these are formed by repeating units of simple sugars. That means what? Carbohydrates have polymers, got it? Proteins are made up of amino acids, isn't it? So amino acids are joined together by forming a peptide bond. Yep, the reaction for peptide bond formation is a condensation reaction. So the two amino acid molecules, they come together, lose a water molecule and form a peptide bond, correct? Even in carbohydrates, when the simple sugars are coming together, okay, when the simple sugars are coming together, what's happening? These simple sugars lose a water molecule, and ultimately what happens is a glycosidic bond is formed, okay? And this glycosidic bond that is formed is responsible for joining the simple sugars together to form the carbohydrate polymer, okay? In proteins, amino acids are joined together by peptide bond, yeah, by peptide bond, and ultimately what you get are polypeptides. So polypeptides are polymers again. Nucleic acids have the building blocks called nucleotides. So even if we talk about RNA and or DNA, both of these are what? Both of these are polymers of nucleotides, right? But then what are lipids? Lipids, if you can recall, are esters of fatty acids and glycerol, right? So we have the glycerol molecule, yep, yeah, and then we have the fatty acid chain, right? Okay, what do we have? We have the fatty acid chain. Yeah. So when one fatty acid is joined to the glycerol molecule, it's called as a monoglyceride. When two of them are joined to the fatty acid molecule, it's called glycerol molecule, it's called as a diglyceride. And when three, we have triglycerides. Now there are certain special types of lipids which are called as phospholipids, isn't it? So so it's a diglyceride with a phosphate group, yep, attached. So lipids are not polymers. Okay, lipids are simple structural compounds. Yeah, all the others are polymers. Attractants and rewards are required for entomophily, hydrophily, cleistogamy, and anemophily. Entomophily is the pollination by insects, isn't it? Hydrophily is the pollination by water. Anemophily is the pollination by wind. And what is cleistogamy? Cleistogamy is the process where the flowers don't open, right? So the flowers are closed, and this leads to self-pollination. So obviously in cleistogamy, self-pollination is occurring. Now hydrophily is water pollination, anemophily is wind pollination, correct? Entomophily is where where the nature is utilizing insects for pollination. Now when the insects are utilized for pollination, there are certain rewards that are supposed to be given to the insects. And what are these rewards? These rewards are fragrance, nectar, yep, large attractive petals. Okay, large attractive petals and fragrance help attract the insects. Nectar is the reward that they get. Another reward that is utilized by certain insects such as bees is pollen. Yeah, bees also utilize pollen. So all of these are rewards. Now these rewards are not required for hydrophily and anemophily. Why? Because it's just wind and water pollination. So if we look at the plants which are pollinated by water or wind, they do not produce large flowers. Their flowers are small, inconspicuous, and they are dull colored. Okay, but what other adaptations do they have is they have versatile anthers, etc., so that the pollen grains can be carried away easily by the wind. Whereas entomophily, ornithophily, which is bird pollination, is considered, then certain rewards are supposed to be given, and these are these rewards. But one thing that you need to remember as a distinguishing factor for ornithophily is that birds have a poor sense of smell. So that is the reason the plants that use ornithophily, the plants that are ornithophilous plants, they won't be wasting their energy in creating fragrance. Okay, so the answer over here is what? Entomophily. Correct? Next, receptor sites for neurotransmitters are present on. Now here what we are talking about is synapse. Okay, now synapse. Now when we have a neuron, at the end of the neuron what we have is the synaptic knob. Okay, so when the neurons are connected, yeah, and this is the dendrite of another neuron, correct? Okay, this is what? This is the dendrite of the other neuron. So this part, this junction is known as what? Synapse, right? This junction is known as what? Synapse. Now when we zoom into the synapse, what do we see? We see a presynaptic neuron, and then we see the postsynaptic neuron, correct? I'll just draw it a bit bigger. Okay, so this is the synaptic knob, and this here is the dendrite of the subsequent neuron, correct? It's the dendrite of the subsequent neuron. And this here is the synaptic cleft, correct? Which is a gap of about 2 to 20 nanometers between the two neurons. So this neuron over here will be called as the presynaptic neuron. Okay, and this one over here is called as what? The postsynaptic neuron, right? It's called as what? The postsynaptic neuron. Now, presynaptic neuron, postsynaptic neuron. Okay, now what happens over here is the presynaptic neuron has these vesicles which contain the neurotransmitter, right? And it has calcium channels. Okay, calcium is present in the surrounding region of the neurons. So when the neuron receives a signal, or when the electrical impulse is passing through the neuron, when it reaches the synaptic knob, the calcium channels open and calcium influx takes place in the presynaptic neuron. Okay, now as calcium influx takes place in the presynaptic neuron, excuse me, what happens over here is these calcium ions, they push or they bring about the joining of the vesicles to the membrane of the presynaptic neuron. And when this joining takes place, what basically happens over here is the neurotransmitter vesicle bursts open in the synaptic cleft and the neurotransmitter is released into the synaptic cleft. And on the postsynaptic neuron, that is the dendrite of the subsequent neuron, there are receptors. So these neurotransmitters will now be released into the synaptic cleft and they will be received by the receptors on the postsynaptic neuron, and ultimately a new impulse will be generated in the subsequent neuron, which will be taken ahead as the nerve impulse, and action or a reaction will be brought about for the stimulus. Got it? So receptor sites for neurotransmitters are present where? Postsynaptic neuronal membranes, right? Moving ahead with the next question. Adult human RBCs are enucleated. Which of the following statements are the most appropriate explanation for this feature? Now, see, the most appropriate answer over here is only the fourth statement. Yep. Why? Because the RBCs are enucleated. Yep. Enucleate means what? Their nucleus is removed. So when the RBC progenitor cells, that is the cells from which RBCs are born, when the RBC progenitor cells come into picture, when it gives rise to RBCs, there is one RBC which retains the nucleus and the other RBC that is devoid of the nucleus. So the RBC that retains the nucleus is killed by phagocytosis, okay? Whereas the RBC which is devoid of the nucleus is selected as the functional RBC. It's not just the nucleus that is devoid, the RBCs also not have the mitochondria, okay? Simple things. Eukaryotic nucleus occupies a very large space, okay, in the eukaryotic cell. Apart from that, if mitochondria are present, let's see. The function of RBCs is what? The function of RBCs is oxygen transport. And for this to be achieved, there is hemoglobin present. So if mitochondria are present in the RBCs, wouldn't the RBCs utilize the oxygen themselves for the purpose of aerobic respiration? They would. So in order to avoid this, mitochondria are not present in the RBCs. Another thing, nucleus is also removed to make as much space possible for hemoglobin molecules in the RBCs, okay? So to increase the surface area or available area for oxygen transportation by packing as many hemoglobin molecules in the RBC as possible, the body, the blood system ensures absolutely perfect oxygen transportation throughout the body. Yep. So all their internal space is available for oxygen transport. Every single RBC, okay, every single RBC on an average contains about 200 million molecules of hemoglobin, okay, single RBC. And every hemoglobin molecule has the capacity to carry 4 oxygen molecules. So imagine the rate at which RBCs are providing oxygen to our body. Yep. Next question. Artificial selection to obtain cows yielding higher milk output represents. Now this question combines 2 chapters from 12th grade, that is strategies for enhancement in food production and evolution. Okay. So to understand this, let us understand the concept from strategies for enhancement in food production, and then we'll move ahead with the evolution, this thing. So this process is actually called as what? Animal breeding. Okay. Animal breeding has 2 types, inbreeding and outbreeding. Okay. Now what is a breed? The animals which show similar characteristics have a common ancestor for about 4 to 6 generations. Yep, they show similar characteristics, genetic features, appearances, etc. are termed as what? breeds. Okay, so there are different breeds of cows. Okay, so cow's scientific name is Bos indicus. Okay, and we have different types of cow breeds, and these different types of cow breeds are utilized for animal breeding experimentation. Now, how are they utilized? See, the cows, what are we looking at, are milch animals. Okay, milch animals means what? The animals that are capable of producing milk. So the cow that provides highest possible lactation, okay, is the one that will be selected. So on a daily basis, if a cow is capable of giving about 15 to 20 liters of milk on a daily basis, then that's a high lactation cow. Okay, now such a cow will be selected in animal breeding. Okay, so what are we doing over here? The selectors or the people who are carrying out breeding are selecting one characteristic, isn't it? That is high milk yield. They're selecting one characteristic, that is high milk yield, and ensuring that this particular characteristic is kept in the breed. So it is either maintained or it is enhanced. Okay, so ultimately the character is being pushed for the selection of only one extreme character, which will be high milk yield, right? So ultimately this type of selection will give us directional, isn't it? In evolution, in earlier practice sessions also, we studied that there are three types of selections: disruptive, stabilizing, and directional, isn't it? So stabilizing is where an intermediate quality is selected and it is kept. Okay, directional is like when you have two extreme qualities, one of the extreme qualities is selected and the curve is pushed in that particular direction. Disruptive is when both the extreme qualities are selected depending on where the organism is dwelling, what type of adaptation the organism requires. Yep. Next, good vision depends on adequate intake of carotene-rich food. Select the best option from the following statements. What are we talking about over here? We are talking about a component which is called as rhodopsin. Rhodopsin is a pigment that is found in the rods of the retina, isn't it? Yeah, it's found in the rods of the retina. Now this component is also known as what? Visual purple. Yep. Now this visual purple or rhodopsin has two components in its chemical structure: retinol, which is an aldehyde derivative of vitamin A, and another component called opsin. Okay, so these two components are the ones that form the visual purple, right? And when light falls on it, there is dissociation of these two components from each other, ultimately resulting in the generation of action potentials inside the cells of the retina, the receptor cells of the retina, which will be detected in the visual cortex as the image. So retinol is a derivative of vitamin A, which is retinol, right? So retinol, the retinol is obtained from pre-vitamin, okay, pre-vitamin A molecule, pro-vitamin A molecule, sorry, which is called as beta carotene. Yeah, which is called as what? Beta carotene. So vitamin A derivatives are formed from carotene. The photopigments are embedded in the membrane discs of the inner segment. Retinol is a derivative of vitamin A. Retinol is a light absorbing part of all the visual photopigments. What will be the answer over here? Yep. So this will be the particular answer. Got it? 1, 3 and 4. Next, the genotypes of husband and wife are IA, IB and IA small I. Correct? Among the blood types of their children, how many different genotypes and phenotypes are possible? Simple enough, a quick Punnett square, right? Got it? Remember that small I is the recessive one, double recessive, that is homozygous recessive condition will give you blood group O. Otherwise, heterozygous condition will depend on what the corresponding dominant allele is. So what am I getting over here? So how many independent genotypes do we have? Homozygous dominant A, then AB, heterozygous A and heterozygous B. So we have 4 genotypes. But how many phenotypes do we have over here? Blood group A, isn't it? Blood group AB and blood group B. So we have 4 genotypes and 3 phenotypes. Right? Yeah, simple Punnett square, a quick evaluation and you get your answer. Root hairs develop from the region of elongation, root cap, meristematic activity and maturation. The chapter that we are talking about over here is morphology of flowering plants, isn't it? So what do we have over here is, we have, if we look at the root tip, so if this is a tap root that we are talking about, yeah. So if we look at this particular part under the microscope, what do we see is the root tip with the root cap, right? So here this is the root cap. At the tip of the roots and at the tip of the shoots, what do we have? Apical meristems, isn't it? And what is the function of apical meristems? Function of apical meristems is multiplication and growth. What type of growth? Primary growth, isn't it? Growth in length, upwards and downwards. So apical meristems will be what? A region of meristematic activity, isn't it? Meristematic activity. Now, as the meristematic activity is taking place, what's happening? More and more cells are getting added in the region just behind the region of meristematic activity. So this over here becomes the region of elongation, right? Why? Because that particular region is getting more and more new cells and it is getting elongated. So that's the region of elongation. Now, when do we say that a cell has matured? We say that the cell has matured when it has attained its functionality, right? It has become completely functional. That is the time when we say that the plant cell has matured. So if the function of the root is absorption of water and minerals, so for that reason, what does it require is the root hair. So when the cells give rise to root hairs in all the directions, yeah, these cells are said to be matured. That is the reason the region where the root hairs are seen is known as the region of maturation. Right? Got it? So where will you find root hairs? You will find root hairs in the region of maturation. Got it? Simple enough? Great. A disease caused by an autosomal primary non-disjunction. The answer is literally given, I mean, there's nothing difficult about this question, but let's just look at the genetics of it. What is non-disjunction? See, we have the monad chromosomes. Yeah, This is the diploid condition, isn't it? So we have the monad chromosomes. Now after this S phase, these monad chromosomes become the dyad. Yeah, these become what? The dyad. So what has happened over here is, see, remember that the number of chromosomes, when you count the number of chromosomes, count the centromeres, not the sister not the chromatids. So this chromosome number is 2. This chromosome number is still 2, because what you are seeing are 2 centromeres. But because of S phase, the amount of DNA is doubled, right? The amount of DNA is doubled. So for example, if there was 20 picograms of DNA over here, there's 40 picograms of DNA over here. But has the chromosome number changed? The chromosome number hasn't changed, correct? Now what will happen? When mitosis will take place, there will be 2 cells that will be formed, which will now receive 2 chromosomes each. So why has this happened? Because disjunction takes place. Disjunction is what? Centromere is a junction where the sister chromatids are joined. Disjunction is the breaking of that junction, right? So when disjunction takes place, there is segregation of chromosomes taking place. So the chromosomes can be chromosomes can be distributed to whatever the daughter cells are, correct? But what is the phenomenon? The phenomenon is called as non-disjunction, what is being talked over here. Now what is non-disjunction? So if non-disjunction is occurring, initially this will be the condition in one cell and this will be the condition in other cell. So this particular cell, let me rename it as A and B. So the cell named B has 1 less chromosome, and this particular chromosome will now be represented as this, right? And cell A has 1 extra chromosome, so that is trisomy, isn't it? So this is trisomy and this is monosomy, correct? Now here they are asking autosomal. So recall, when we talk about chromosomes, we have autosomes and allosomes. Allosomes are sex chromosomes. So any problem with the X and Y chromosome will be an allosomal condition. Apart from that, it will be autosomal. So Klinefelter's is what? 44 plus XXY, right? What is Turner's? 44 plus X0, right? So both of these are allosomal conditions. Sickle cell anemia is a mutation, right? It's a mutation, it's a transition mutation, whereas Down syndrome is trisomy 21. That is an autosomal condition. So what's my answer over here? Non-disjunction of the 21st chromosome during mitosis is going to give us Down syndrome. The final proof for DNA as the genetic material came from the experiments of Hershey and Chase, Avery McLeod McCarthy, Hargobin Khurana and Griffith. Now, these were the people who carried out extensive research in molecular biology to determine the genetic composition of many things. So, first and foremost thing, Griffith, Avery McLeod McCarthy and Hershey Chase. Griffith gave us the transformation principle. Yeah. So what did he do? His experiment was pretty simple. He studied Diplococcus pneumoniae, which is an organism that causes pneumonia. It's in the name there, right? Pneumoniae. It has 2 strains, R strain and S strain. Okay. The R strain is the rough strain. Now, why is it called as a rough strain? Because it does not have a polysaccharide layer on it. It does not have a polysaccharide capsule. And since it does not have a polysaccharide capsule, that polysaccharide capsule is what is responsible for giving virulence. Okay, so R strain is non-pathogenic. Okay, S strain has a polysaccharide capsule on itself, and this polysaccharide capsule is what makes the S strain pathogenic. So what did Griffith do? Griffith took the R strains and S strains and he worked on them with the help of a mouse. Okay, so he injected the R strain in the mouse and found out that the R strain does not cause pneumonia. Okay, then he injected the mouse with S strain and saw that the mouse developed pneumonia and died. Okay, now the next thing that he did was he took the S strain and killed it. He heat killed it. Okay, and when he heat killed the S strain, he injected the heat killed S strain into the mouse and saw that the mouse is not developing pneumonia and the mouse lived. Next thing what he did was he mixed the heat killed S strain, okay, with living R type, and then he injected the mixture into the mouse and the mouse developed pneumonia and died. So what Griffith put forth was something was released by the dead S strain, which converted the R strain into live S strain, which caused pneumonia and the mouse died. Okay, so Griffith simply gave us a transforming principle. He did not know the biochemical nature of the transforming principle, nothing. Every McLeod McCarthy, what they did was they carried out experimentation around 16 years after Griffith's experiment, okay, where instead of using the mouse, they used cell lysate. So what did they do is they took the S strain, killed it, destroyed the cells, isolated the substances, and started mixing that with live strains and then seeing whether transformation is inhibited or transformation is promoted. So they utilized enzymes. They utilized RNAs, which destroyed RNA, okay, but transformation still occurred. They utilized protease. Yeah, it was a concoction, it was a mixture. They broke down the cell and whatever came out, they used it. So they used RNAs, which destroyed RNA, transformation still occurred. They utilized protease, which destroys proteins, and saw that transformation still occurred. They utilized lipase, destroyed lipids, still transformation occurred. They removed carbohydrates, transformation still occurred. When did it get inhibited is when they used DNAs. Yeah. So with every McLeod McCarthy's experiment, it was established that the transforming principle is DNA. Understand the difference between the results of the experiments. Every McLeod McCarthy's experiment stated that the transforming principle is DNA. It still hasn't established that the genetic material is DNA. Okay, this was done by Hershey and Chase. Okay, so how did they do that? They utilized bacteriophages. It is known that bacteriophages' genetic material is DNA. It injects its DNA, it injects its genetic material, that is DNA, into the bacterial cell. So what Hershey and Chase did is, Hershey and Chase used two types of bacteriophages. One with radioactive sulfur and the other with radioactive phosphorus. So radioactive sulfur will be present in the protein coat. So if this is the bacteriophage, yeah, this is the protein coat. This is where you will have radioactive sulfur. And they used another bacteriophage in which the DNA that is present inside, okay, the DNA that is present inside has phosphorus. radioactive phosphorus on it, correct? So there were two different bacteriophages. Now what they did was they allowed these bacteriophages to infect E. coli. And when E. coli was infected, they checked which part of the centrifugate. So they centrifuged it to separate the ghosts. Now what are ghosts? Ghosts are the empty viral particles. Yep, when the DNA is gone, they're called as ghosts. So to separate the ghosts. Now the ghosts, when you centrifuge it, so here you get the pellet, which is the bacterial cell, and this is where you get the ghost particles of the viruses. Yep, the viral ghost particles. Now what happened over here is, when the centrifugate was studied with this particular bacteriophage, the radioactivity was found in the supernatant, is the liquid. Yep. And with phosphorus, radioactivity was seen in the pellet. That means the DNA had entered the cells, and that is the reason the radioactivity was in the pellets. And that is how Hershey and Chase established that DNA is the genetic material. So understand the differences, okay? Griffith gave the transforming principle. Every McLeod McCarthy established the nature of the transforming principle, that is DNA. Hershey and Chase established that genetic material is DNA. Hargobind Singh Khurana was awarded a Nobel Prize with two others for cracking the genetic code. Okay, so the genetic code table is a work of him and his colleagues. Next, which of the following components provides sticky character to the bacterial cell? The bacterial cell has cell envelope, isn't it? And this cell envelope has three things: glycocalyx, cell wall, and the cell membrane. Glycocalyx is made up of polysaccharide. Cell wall, bacterial cell wall, is peptidoglycan. And cell membrane is phospholipid. Yeah. Glycocalyx, the polysaccharide, is responsible for adhesive properties to the bacterial cell. And hence, which of the following components provides sticky character to the bacterial cell? The answer will be glycocalyx. Glycocarbohydrate. Calyx is a protective covering. Got it? Yep. Great. Lungs are made up of air-filled sacs, the alveoli. They do not collapse even after forceful expiration because of what? The residual volume, isn't it? So even after forceful expiration, there is some amount of air that is present in the lungs, and this air keeps the lungs inflated. Okay, it does not allow the lungs to collapse, it keeps the lungs inflated, and this is known as what? Residual volume. So residual volume generally does not come out of the come out of the lungs. So even if you forcefully expire. Oh, interesting question. If there are 999 bases in an RNA that codes for a protein with 333 amino acids, and the base at the position 901 is deleted such that the length of the RNA becomes 998 bases, how many codons will be altered? Okay, great. So the calculation is absolutely simple. Yeah, what you need to do is, how many nucleotide bases are required for are present in a codon? 3, correct? 901, right? So if I divide 900 by 3, I get how many amino acids? 300, correct? So that means my 300 amino acids are sure that they are going to be formed correctly. The 300 amino acid polypeptide is going to be correct. Deletion is occurring at the 901st base, correct? And how many total amino acids are getting added? 333. So if 300 are getting added properly, then what problem will be seen in how many amino acid residues? 33. Simple enough. Yeah, no high fun math required over here. Simple calculations. A dioecious flowering plant prevents both autogamy, gytonogamy. Okay, so we're talking about pollination, isn't it? Yeah, we're talking about pollination over here. So when we talk about pollination, we have self-pollination and cross-pollination. Self-pollination has autogamy and gytonogamy. Cross-pollination is xenogamy. Okay. Now, autogamy is true self-pollination, that is, the pollination is occurring in the same flower. Gytonogamy is genetically self-pollination, ecologically cross-pollination. That means what? It is the pollination that is happening between two flowers, but two separate flowers, but on the same plant. Xenogamy is absolutely true cross-pollination, where the pollination is occurring between excuse me, two different flowers on two different plants, but of the same species. Got it? So, autogamy, gytonogamy. Now there are two types of plants, monoecious and dioecious. Okay, both of these conditions are seen in unisexuality of flowers. Okay, so monoecious is a condition where you will have male flower, that is the staminate flower, and the female flower, which is the pistillate flower, on the same plant, like in Siamese. Yeah, you have the male flower at the top, male inflorescence is at the top, and the female inflorescence is at the lower levels. Okay, so here what we have is Siamese. Okay, so monoecious condition. Dioecious condition is where you have two different plants altogether, okay, which show you either the male flower or the female flower. Yep. So monoecious condition, even though it does not give you xenogamy, it gives you, yep, got it? I mean, it gives you gytonogamy, not autogamy. Dioecious plants is going to give you true xenogamy, right? It cannot give you autogamy, neither can it give you gytonogamy. So autogamy, gytonogamy, both are prevented when we talk about a dioecious flowering plant. Next, which statement is wrong for Krebs cycle? There is one point in the cycle where FAD plus is reduced to FADH2. Is it true? It's true, isn't it? Yeah. During the conversion of succinyl-CoA to succinic acid, a molecule of GTP is synthesized. Yeah, this is substrate level phosphorylation. What is substrate level phosphorylation? Substrate level phosphorylation is the type of phosphorylation or ATP synthesis that occurs outside or apart from the oxidative phosphorylation that is happening on the inner mitochondrial membrane. So substrate level phosphorylation, GTP is formed, but since GTP is unstable, the phosphate is transferred to ADP and ATP is formed. Okay, so this is substrate level phosphorylation. That is true. Then the cycle starts with condensation of acetyl group with pyruvic acid to yield citric acid. Is that true? That is wrong, isn't it? Yeah, And there are 3 points in the cycle where NAD plus is reduced to NADH2. Yeah, those are the cases. So let's understand this. So glycolysis gives us pyruvic acid. Pyruvic acid undergoes link reaction excuse me, and gives us acetyl-CoA. Acetyl-CoA goes to the mitochondria where now pyruvic acid is a 3-carbon compound, okay? Acetyl-CoA is a 2-carbon compound because during link reaction decarboxylation takes place. Yeah, one carbon dioxide is released. Acetyl-CoA is a 2-carbon compound which goes to the mitochondria, and there it is taken up by oxaloacetic acid, which is a 4-carbon compound, which takes up the acetyl and gives rise to citric acid, which is a 6-carbon compound. Got it? Citric acid is a tricarboxylic acid, right? And since it's the first stable product of the cycle is called as tricarboxylic acid cycle or the citric acid cycle or Krebs cycle because it was discovered by Sir Hans Krebs. Got it? Yep, wonderful. So this is how it works. Phosphoenol pyruvate, PEP, is the primary CO2 acceptor in ribulose bisphosphate carboxylase oxygenase, the most abundant enzyme in the living systems. Okay, ribisco is a carboxylase as well as an oxygenase. When it is a carboxylase, it takes up carbon dioxide and gives it to ribulose bisphosphate, which is a 5-carbon compound. Yep, carbon dioxide, another 1-carbon compound, gives us a 6-carbon unstable keto acid, which breaks down to phosphoglyceric acid, which is a 3-carbon compound. Yeah, and Calvin cycle proceeds to glucose formation. When does it act as an oxygenase? It acts as an oxygenase when carbon dioxide concentrations are low, which is a very common condition in tropical regions, isn't it? Yeah. When the carbon dioxide concentration is low, ribisco is going to act as an oxygenase. And when it's acting as an oxygenase, it is going to be responsible for photorespiration. And photorespiration will result in the removal of about 25% of already fixed CO2 from the plant. So it's a detrimental process for plants. In C3 plants, it is a beneficial process because photorespiration prevents the prevents the breakdown of the light complexes. Okay, so this is what happens over here. So for some plants, it is detrimental. Now, the plants that are growing in low concentrations of carbon dioxide have evolved a mechanism wherein they do not allow the ribisco to come in contact with the air. So they have what? They have bundle sheath cells. So you have the vascular bundles over here. They have the bundle sheath cells, okay, which are agranal chloroplasts, okay, which have agranal chloroplasts, agranal chloroplasts, that is, they do not have grana. And they have mesophyll cells around it, which have granule chloroplasts. Yep. Recall that the enzymes for dark reaction are present in the stroma. So the granule chloroplasts are abundant in grana, whereas the agranal chloroplasts do not have grana at all. So the bundle sheath cells will be the site of dark reactions or light independent reactions, majorly the Calvin cycle. So in the granule chloroplast, we have phosphoenol pyruvate, or phosphoenol pyruvic acid, which is a 3-carbon compound. This is, where is this happening? This is happening in the mesophyll cells in the granule chloroplast, correct? So phosphoenol pyruvate absorbs carbon dioxide and forms oxaloacetic acid, okay, which is a 4-carbon compound. This oxaloacetic acid forms malic acid, which is another 4-carbon compound. This malic acid is transported to bundle sheath cells, where it loses a carbon dioxide molecule, and forms pyruvic acid, yeah, which is a 3-carbon compound. The pyruvic acid is transported back to the mesophyll cells, okay, pyruvic acid. It undergoes phosphorylation by ATP and forms phosphoenol pyruvic acid. This CO2 in the bundle sheath cells, okay, will now enter Krebs cycle or C3 pathway. Got it? Now the first stable compound of this reaction is a 4-carbon compound, oxaloacetic acid. Hence this pathway is known as the C4 pathway. Got it? So phosphoenol pyruvate will be the primary CO2 acceptor in C4 plants. Got it? What is the criterion for DNA fragments to move on agarogel during electrophoresis? Electrophoresis is the process which results in the separation of separation of charged components under the supply of voltage, okay. The movement is based on charge to mass ratio, yeah. That means, so electro is electricity, phoresis means being carried. So something is carrying these molecules away. And what is that? That's the charge to mass ratio which is doing that, okay. So now the charge on all the DNA molecules is what? Negative, because of the phosphate groups, correct? Now the mass is what is going to be the defining factor. So phoretic mobility is inversely proportional to the mass of the DNA. That means heavier the DNA molecule, slower will be its movement in the gel, right? So ultimately what does that mean? Smaller the DNA molecule, farther it will move away or travel in the DNA, in the agarose, sorry. Smaller the fragment size, the farther it moves. Frog's heart, when taken out of the body, continues to beat for some time. Select the best option. Let's just jump on the answer over here. Heart is myogenic and autoexcitable. What do you mean by myogenic? Genic, genesis of something, formation of something. But formation of what here? Formation of the signal for the heart to beat. There are 2 types of hearts, neurogenic and myogenic hearts. Neurogenic hearts are the ones that receive signal for beating from the nervous system. Myogenic hearts are the ones that receive the signal from the muscles. And autoexcitable hearts are the ones that generate their own muscle signal for the heart to contract, okay. All vertebrate hearts are myogenic, autoexcitable. Remember that, right? So same thing goes for frog. Homozygous pure lines in cattle can be obtained by, we were just discussing animal breeding, isn't it? So out of that, if we look at inbreeding, inbreeding means what? It is the breeding of animals that belong to the same breed. It's the mating of animals that belong to the same breed. Now what does this mean? These animals should have a common ancestor that goes back to about 4 to 6 generations. That is when they will be called as the related individuals. So it's the mating of related animals, Yeah, because of which what happens is pure lines are maintained. So homozygosity sets in with respect to the alleles and pure lines are formed in inbreeding. But one thing we need to remember is that continuous inbreeding leads to something which is called as inbreeding depression. Yeah, and this inbreeding depression leads to the loss of fertility, okay, and survival characteristic. Yeah, so this is basically what happens in inbreeding depression. To overcome inbreeding depression, one single outcross can be performed. So it's the mating between individuals of the same breed but without a common ancestor for 4 to 6 generations, that is outcross. Got it? So inbreeding is what is going to be helpful. So mating of related individuals of same breed will help in this thing of to give rise to pure lines by maintaining homozygosity. Got it? Anaphase promoting complexes is a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in human cell, which of the following is expected to occur? It's anaphase promoting complex, isn't it? Since it is anaphase promoting complex, it is going to promote what? The disjunction of sister chromatids. If APC is defective, will disjunction take place? No. If disjunction is not taking place, what will happen? Chromosomes will not segregate. We just discussed that. Yeah. Next, last question for today. Spliceosomes are not found in cells of fungi, animals, bacteria, plants. Spliceosomes. What are spliceosomes? Recall a process called as exon splicing. Exon splicing is one of the post-transcriptional modifications, and post-transcriptional modification is a characteristic of what? Eukaryotic cells, isn't it? Yeah. Now, which of these are eukaryotes? Fungi, animals, plants. So spliceosomes, which carry out exon splicing, it's a group of proteins that carry out exon splicing. So if they are carrying out exon splicing, that means it's a process that is taking place in the fungi. In the eukaryotic cells. Bacterial cells have coupled transcription translation. They don't have post-transcriptional modifications. Got it? Yep. Great. So this was for today. Yeah, we have one more session, and then I'll see you in the next session. Okay. Bye guys. Have a nice day.